In the name of Allah, the most beneficent and the most merciful. Dear students, Assalamu alaikum wa rahmatullahi wa barakatuhu. I hope you all are fine and well. Dear students, I welcome you in Pakistan International School Thais Virtual Learning System. Today we are studying biology in this lecture, and this is biology for a class for grade 9th junior. Dear students, we are gonna start our new chapter with the new topic, that is chapter number 5, that is about cell cycle. This is very important topic, very important chapter as well, because we can say that without this chapter, biology is nothing. Because this chapter is about cell division. How the cell will divide and how it is going to prepare for the division, we will see in this chapter. What is the significant role of the division, why the cell divides, and what are the steps, and what is the usage of that in our daily life. We will talk all about this in this chapter. Inshallah, you will find it very interesting and informative, because lots of things uh, are there which you didn't hear before. So let's get started. Today's topic is interface. What is interface? We will see right now. So Bismillahirrahmanirrahim, we will start chapter number 5, that is about cell cycle. If you see at the background, there is a cell, eukaryotic cell. So how the cell divides, we will see all this in this chapter. As you know that Rudolf Virchow has given one statement which we have seen in chapter number 4 in the start, that was present in the cell theory, the third statement of cell theory. What is that? Omnis cellulae, e cellulae. Like the new cells arise from the existing cells. So how the existing cells, the old cells divide to the new cells, this is actually the basic of the life. This is the basic of, we can say, the survival of the species. This is the basic of surviving of any species on the earth. Okay, so we are gonna see here see at the background, this is the cell, this is eukaryotic cell, and you can see different organelles in that, and here you are looking at cell cycle. So you will be confused what is cell cycle actually. So you can see here there is a cycle. Cycle means the process which are repeating again and again. The blue one, you can see the interphase. This is the longer phase, and the green one is mitosis, then cytokinesis, then the cell divides into two, then this cycle repeats again. And in the interphase there is G1S, G2 phase for the preparation of the division of the cells. So let's talk about cell cycle. Actually, it is a series of events from the time a cell is produced until it completes mitosis and produces new cells. So it carries two phases. This whole circle seems to be a cycle. It is represented in the cycle because it repeats again and again. This portion, this green phase, this will come after this blue phase. So this blue phase indicates the phase interphase. So this is the cell cycle in which two main phases are there, interphase and mitosis, M phase. Inter means between, phase means the time, the stage of the division of the cell cycle. So this is in between stage between two mitotic division. One the cell do division, then it will go through interphase, then it will again divide. So this is that's why it's called interphase. It is the longer phase, as we have seen that interphase is the longer phase and cell prepares itself for the cell division here. And in mitotic phase, that is called M phase, this is the shorter phase and this is called the dividing phase as well. Like after this, the cell divides into two. So in interphase, we are having some other phases also. We will discuss in detail right now in next slides. So you can see here the changing in the cells like this and like this. What are these changing, in which stages they are going on? You can see here the growth and DNA replication, growth and final preparation for division. So we'll talk all about that. And here if you can see there is M phase. In M phase, early prophase, this is a four main stages of mitosis, but it can be divided into early and late prophase, metaphase, anaphase, telophase, and cytokinesis. What are these? We'll discuss in the next lecture, inshallah. But right now we are going to talk about the very main phase of the cell cycle, that is interphase. So interphase is the time when a cell's metabolic activity is very high, and in that cell performs various functions at a time. So three main phases of interphase is there. Like when, for example, this is at the background, there is eukaryotic cells. So eukaryotic cells, this is mature cell now. So before going to divide, it will prepare itself. When it will prepare itself, so what changes are going on in the cell, that is called interphase stage. And definitely it has to prepare, like we have to go to the party, so we take a lot of time for our preparation, and we do lots of things, dressing ourselves. So like that, cell also prepares itself before division. So it is composed of three main stages. This is called G1 phase. G1 is said to be gap 1 phase. Then S phase, that is called synthesis phase. Then G2 phase, that is called gap 2 or gap 2nd phase. Then there is G0 or G0 phase. So we consider three main phases because G0 phase is out of the cell. So here sometimes the cell divides, sometimes not, sometimes the cells don't divide so quickly. The process is very slow. So we'll talk about in the last, inshallah. So here you can see the interphase stage. This blue one is interphase. This up to here. This is mitotic phase. So in this, the longest phase of the interphase is G1 phase, in which the cell is going to prepare and forming lots of things, then S phase, and then synthesis phase. S is the basic phase, G1 is the longest phase, and G2 is preparation for mitotic division. So let's see the phases of interphase. So the number one is G1 phase. Gap 1, it means before synthesis and mitosis phase. there is the gap phase in which cell does not rest. Actually, it prepares lots of metabolic activities. And here what is going to happen, the cell increases its supply of protein. For supply of protein, what organelle is needed? Yes, that is ribosomes. So ribosomes are required for what? For protein synthesis. So when there is increase in supply of proteins in the cell, so definitely cell will increase the number of ribosomes. Okay, as well as because it needs its metabolic activity is too high, so it's need energy. So for the production of energy, mitochondria is required. For respiration, because due to respiration energy is produced. So for energy production, more energy production, mitochondria is required. So for more energy production, more mitochondria has to be synthesized. So cell increases the number of mitochondria, ribosomes, Golgi apparatus, and all the organelles which are required for the next phases. When these organelles are increased in numbers, so it increases in the size as well. So the cell grows in size because all organelles cannot be accumulated in the specific. So cell membrane we have studied before, it is a little elastic, so it increases the size. Then synthesis of various enzymes that are required in the next phase, that is done here. Which is the next phase? That is S phase. So synthesis of various enzymes. Enzymes are also what? These are proteins. So it means when the protein is increased, so ribosomes are increased. So when proteins increase, enzymes will also be increased because there is more energy production, more respiration, more protein synthesis. For these two processes also, the cells requires more enzymes because cells cannot perform their function without the enzymes. Enzymes are actually chemicals which are protein in nature. So we will talk about the enzymes in chapter number 6, inshallah, the next chapter, but in 9th senior as well as in chapter number 7, we'll study about the importance of enzymes, how they're used in photosynthesis and respiration. So enzymes are required for these two processes also, for energy production, for respiration, for ribosomes activity like for protein synthesis, as well as the enzymes which are required in S phase, which is going to be done in X phase. We will S phase we'll talk about in the next slide. So for in S phase, main process is the duplication of enzymes like chromosomes. It means the DNA which is present as thread-like structures in the nucleus. Here we can see this is mitotic phase. So we are talking about this interphase. So this is G1 phase, the longer phase, and here we can see these are thread-like chromosomes inside the, this is thread-like chromatin material in the nucleus. So it has to be duplicated. When it has to be duplicated, it will become chromosome structure like this. So this will become duplicated in S phase, but before preparation for S phase, that is done in G1 phase in the form of synthesis of lots of enzymes for S phase. Okay, so S phase, as for synthesis, synthesis means for product, synthesis means production or formation or making, production. Okay, production or making. So anything which is going to be formed like photosynthesis, like preparation with the help of photo or light. So that is preparation, production, and making any type we can see. So synthesis phase, what is done in synthesis phase, like new chromosomes are formed. Not new actually. The old one, here you can see in the synthesis phase, these are the old one and they are duplicated in the next copy. Like they are going to be copied here. Like how many chromosomes are present in human being? These are 46. Okay, so 46 chromosomes are duplicated. So when they are duplicated, 46 and 46, how many we will get? Yes, 92 chromosomes. Like if one is having this type of structure, so other will also having this type, the same structure. Actually, this is not the proper form of this one. So like this one will be copied into exact copy uh, and replica of this one. So these chromosomes are duplicated. So it become 92 chromosomes in human beings in one cell before the division. Because when the cells divide, the chromosomes will split and the cells divided into two, so both cells will have same number of chromosomes. So chromosome numbers uh, must not be increased or decreased. If increased or decreased, what will happen, we will see in the next slides, inshallah. So duplicates of chromosomes is done and each chromosome consists of two sister chromatids. Now here is what are the chromatids, what are the sister chromatids? Like this is one chromosome, got it? When it will have its duplicated chromosome like this, so this is called chromatid and this one is called chromatid. Now how many chromatids we are having? 1 and 2, because this is same of this one, so this is called sister chromatid. So each chromosome is having two sister chromatids. Okay, so here we can see the chromosomes or DNA is going to replicate or duplicate and then it will change the form like this and they will become like as chromatin. Now they will become chromosomes. The synthesis phase is very important phase indeed in interphase stage. Now G2 phase, like gap 2 phase. Gap 2 phase means between mitosis and S phase we are having another gap that is not unnecessary, but it prepares, the cell prepares here proteins again, but before the proteins have been formed, enzymes have been formed for the synthesis phase. Now the proteins are formed for mitosis or mitotic phase, because mitotic phase is very near to G phase now. Soon after G2 phase completion, mitosis will be started. So the cell uh, double checks the duplicated chromosomes for errors, making any needed repairs in G2 phase. If here in S phase, when the chromosomes are duplicated, if chromosome sequence is changed, if there is any problem, so in G2 phase there is a check over there, like some enzymes just check whole of the process and they repair the chromosome. That is the natural process. So now cells is going to prepare itself for mitosis or the division. So mainly for production of spindle fibers, we have talked about before that centrioles form spindle fibers. These are the fiber like structures which we'll talk about in mitosis as well. So because spindle fibers are also made up of some chemicals, some proteins, so the cell in G2 phase, those proteins have to be prepared which are required later for the production of spindle fibers. After G2 phase, okay, one more thing here, that when um, like in this interphase stage, not only chromosomes duplicated, but all the required organelles are duplicated. For example, centrioles, they are also duplicated because one centriole has to move on one side of the nucleus. So in G2 phase of interphase, the cell enters the division phase now, that is called M phase after the G2 phase. So that is characterized by the cell division, and after that, after division, cell divides into two cells which are having a double number of chromosomes. What is this? And how the double number of chromosomes are there, we will see inshallah in mitosis. It's so easy and very interesting, inshallah. You will find it interesting when we see mitosis and meiosis. Okay, so here we will talk about the next phase, which is actually not the part of cell cycle, but it is out of that, but it is also very necessary. This is actually the temporarily that is the phase in which the cells which undergo temporarily or permanent stop dividing. So they have entered in G0 phase, but soon after G1 phase. Because here chromosome numbers in S phase are duplicated, if the cells just go in G0 phase, so after S phase, so it means here the chromosome number will be 92 duplicated. So there will be the problem. So that's why here, before duplication of the chromosomes, the cell undergoes this is one chromatid. So when it gets duplicated, it will become two chromatids, as I have shown you before. So it is in one chromatid. It means one chromatid means the chromosome number is 46 here. So after G1 phase, they go under G0 phase, like in the rest phase, in the sleeping mode, because some cells do not divide in our body. What type of cells are these? So in multicellular eukaryotes like human beings, cell enters G0 phase from G1 and stop dividing. Some cells remain G0 phase for indefinite period of time, like the long period of time. It means permanently. So these are neurons, neurons. If you are paralyzed by damaging the neurons in an accident, so they will not be recovered. They can be like progression can be done, but the person cannot be recovered if the neurons are damaged because they don't divide. As well as some cells enter G0 phase or G0 phase semi-permanently, like for some period of time. For example, liver cells do not divide rapidly, but for it's very slow progressive division, and as well as kidney cells don't divide permanently, if like semi-permanently. If they can divide, so why there is need of kidney transplant and liver transplant? That's why they are in semi-permanently phase, just slowly, like after some period of time, they can they just divide, and some cells, not all. There are many cells which do not enter G0 phase or G0 phase. So they continue to divide throughout an organism's life. So they go through the interphase again and again and again and again and again and again. So the cells which are including in those in that type of category, they are epithelial cells, like your skin cells, the skin cells of internal organs, as well as your skin also. So epithelial cells grow and they divide rapidly. So the events of cell cycle are ordered. This is very important thing. Are ordered and directional. Ordered like one after the other. Which phase has to come one after? G0 phase cannot come, never comes after S phase or G2 phase. It will always come after G1 phase. So S phase cannot come after G2. So this is directional and sequential as well. And it is impossible to reverse the cycle. Like if the cell has been gone from G1 to S phase, so it is an S phase to G2 phase, so it is hard, not hard actually, it is impossible to reverse the process in any way, because that's the natural process and controlled by lots of systems of enzymes and different type of things. So uh, here we just got the summary of interphase also. Like it is cell cycle is composed of two phases, interphase and M phase. M phase is cell division phase. Before cell division is shorter, interphase is longer, and in longer phase it is interphase is having three phases: G1, S phase, and G2 phase. And out of the because this helps in division, so because with the cells we don't divide, they go under the sleeping period, temporary permanently stoppage, uh, stop dividing a phase. So here like in G1 phase, synthesis of proteins and organelles. So there is growth of the cell in size, as well as the next one is S phase, where the duplication of chromosomes occurs, as well as where the duplication of chromosomes, cell also grow in size again. Then more growth of the cells, because there is more synthesis of proteins which are essential for mitosis. Now after G2 phase, mitosis is done and cell is divided into two. It means here the 96 how many chromosomes? 92 chromosomes were there. So while after dividing, both cells are having 46 chromosomes. And here the cell is called 2n. 2n means double number of chromosomes. Double number of chromosomes, like 46 is double number of chromosome. Okay. And if we do it half, so that is 23 chromosomes, and that is n number of chromosomes. So you must learn these two terms, you must remember in your mind, because that will help you in mitosis and meiosis understanding. Okay. So in mitosis, when the cell divided into two, the cell will have 46 chromosomes, and in meiosis the cell divided into four, so it will having 23 chromosomes in each cell. So how this all will be done, we will see inshallah. Very interesting topic, mitosis and meiosis, in the next slides, in the next lecture. Okay, that is all for today, my dear students. I hope you understood, found it very interesting and informating, and that is of daily use also. In our body, it's going on every time for whole of the life, somewhere else in the cell, in like sometimes in the blood cells, sometimes in the epithelial cells, sometimes in the eye, sometimes everywhere in the body it's going on. So wish you all the best, my dear students. Best of luck. Inshallah, we will see. And the next topic, mitosis, in the next slides. Allah Hafiz. Assalamu alaikum wa rahmatullahi wa barakatuh.